everybody. So I'm going to talk today about MedGenome's TCR-based neoantigen prediction pipeline and screening solutions for immuno-oncology research. So before I start, a little bit of introduction about who we are and what we do. We are a fairly young company. We were founded in 2013. We are headquartered in Foster City, California. We have global offices in India and Singapore. We are an NGS-based diagnostics company. We are also the founding member of Genome Asia 100K. It's a nonprofit initiative to sequence 100,000 genomes from the Asian subcontinent. As you see here, the Asian, this is the population distribution, and the genome information from the Asian subcontinent is highly underrepresented. With regards to our services, we started off primarily as an NGS company, and then we expanded into different disease areas in the eye, the diabetes, and cancer. Today I'm going to talk about primarily our immunotherapy solutions. One is for vaccine predictions. The second one that we have that I'm not going to talk about today is about the tumor microenvironment, and I'm also going to touch upon our TCR profiling platform. So as Prithvish highlighted that, so there's been a tremendous momentum in the global immuno-oncology trends, and this is just the latest numbers that came out in Nature reviews this month. And it's been shown that there has been a 76% increase in T-cell targeted immune modulators. Those would be PD-1, PD-L1, anti-CTLA-4. There are about 228 active agents that are being tested in the clinics. There's also a tremendous increase in the cancer vaccine space and in the in the cell therapy space, which is mainly TCR therapies and CAR T-cell therapies. The challenge, however, is we still have a long way to go, since most of these treatments are approved only in a small percentage of cancers. And additionally, the overall response rate across all cancers is very poor. And as you heard from the speaker before, the way to improve this is to look for biomarkers of response, one. And some of the examples that are given below is looking at the neoantigen load of the tumor, the immune cell infiltrates in the tumor, and also closely looking at the tumor microenvironment. The second approach would be to start looking into combination therapies, and we are already seeing promising results from the multiple IO drug combinations that are being tested in the clinics. So with regards to increasing response of checkpoint inhibitors, one way to extend their long-term benefit, one way is to look at to break tumor tolerance, and that can be done by either IO drug combinations or cancer vaccines or putting in engineered T cells. My talk today is going to focus on three different areas of this. One is a vaccine prediction algorithm, a cell-based assay to validate those peptide candidates, and then looking at TCR-specific response to those antigens. The second approach to overcome some of these challenges is to develop more robust biomarkers for patient selection. One is to look at the tumor microenvironment, looking at the tumor itself, the tumor stroma, the immune cell compartment, and the second is to profile the tumor microenvironment for T cells with respect to specific TCRs, and then also look for biomarkers of immune-related adverse effects. So MedGenome has developed tools and solutions in all these areas. I'm going to primarily focus on the top three for tonight's talk. So cancer vaccines, how do they? So cancer vaccines arise from somatic mutations that happen in the tumor. So these somatic mutations get transcribed into mRNA and made into proteins. They get proteasomally processed. They bind to TAP, the transporter that transports them into ER. They get loaded onto MHC or HLA and then get presented on the cell surface. This MHC peptide complex is then recognized by the cognate TCR on a T cell. It gets activated, converts the T cell into cytotoxic T lymphocyte that kills the tumor. So in a clinical setting, one would derive cancer vaccines. It's a personalized therapy, so they would mainly come from the patient's tumor, which would be sequenced. The sequencing information would then be channeled into prediction pipelines. Currently, most of the prediction pipelines rely heavily on HLA binding with a cutoff of 500 nanomolar. These peptides that are selected are then screened using cell-based assays, and the final cocktail of peptides are then injected into the patient. So our prediction pipeline is slightly different from what the field currently uses, in the sense that these in silico prediction pipeline recapitulate most of the in vivo processes that take place in formation of the antigen. The key criteria that most people use is binding of HLA to the peptide. So in our pipeline, we have used an additional criteria, which is the interaction of the MHC peptide complex with the TCR. And the reason why we decided to integrate in a TCR interacting component is that when we looked at the positive and negative immunogenic peptides, and this is immunogenic based out of a CD8 T cell activation assay, not a multimer binding. So here you're really capturing a CD8 T cell functional response like production of interferon gamma. So when we look at immunogenic peptides from IDB data sets looking at both immunogenic and non-immunogenic, we see that the enrichment of amino acids at position 2 and 9, which are the amino acids that interact with the HLA molecule, are the same in both immunogenic and non-immunogenic peptides. Additionally, when we plot these non-immunogenic and immunogenic peptides based on their HLA binding, we see that, and keep in mind the cutoff for calling anything immunogenic is less than 500 nanomolar. We see that a large fraction of the immunogenic peptides have a binding affinity of greater than 500 nanomolar, and also a large fraction of the non-immunogenic peptides have a binding affinity of less than 500 nanomolar, meaning if you use HLA binding as a criteria for predicting immunogenicity, you'll end up calling more false negatives and false positives. Now, how do we go about this problem? So based on the crystal structure of MHC peptide TCR complexes, we see that the amino acids 3 through 8 are on the TCR interacting interface. But if you look at their sequence, there is no enrichment of any particular amino acids in this region. So then we decided to look into the physicochemical properties of these amino acids instead of just the hydrophobicity of these amino acids. In order to pull out the physicochemical properties, we started with 1655 IDB peptides and then manually curated the ones that were found to be immunogenic and non-immunogenic in human CD8 T cell activation assays, where they produced interferon. And based on this, we then, across the 9 positions of a 9mer, we collected about 12,000 features from AA index and PepLib descriptors, and an additional few indexes from peptide processing and HLA binding. So when we took all these 12,000 features across all the 9 amino acids and applied it to a known data set, we saw that with increased sensitivity, there was also an increase in false positive rate. So this told us that these large number of features need to be distilled down. So we then used a decision tree-based classifier and further distilled down these features into smaller subsets, and then used an ensemble-based voting mechanism to assign an immune score. So an immune score of greater than 0.5 would be called immunogenic, and an immune score of less than 0.5 would be non-immunogenic. So when we applied this to the data set again, we found 2 groups of features. One group definitely looked better than the one where we did not select for special features. But the second group showed a very high concordance of sensitivity with low false positive rate. So when we then looked at what features we picked in group 2, in the plot you see these were the top features and across the different positions of the amino acids in a 9-mer peptide. The top feature that was highlighted was the helix turn helix feature. And if you look closely in position 2, position 6, and position 8, this feature is highly enriched. And in fact, when you look at the crystal structure of a peptide in complex with TCR and MHC, you can see that position 2 and position 9, that are the anchoring residues of the peptide, and then the peptide kind of makes a turn getting into the TCR groove post position 2. So in order to make that turn, there has to be a kink in position 2, position 6, and position 8, where the peptide kind of arches out of the MHC groove and interacts with the TCR. And indeed, in our predicted algorithm, we see enrichment of these helices at this particular position. So once we saw that, we were pretty confident that we are able to predict an interaction of the MHC peptide complex with the TCR. The next step was to validate the predicted antigens using a cell-based assay. And in order to do that, we used an immune cell culture system where you generate dendritic cells from monocytes, purify T cells from PBMCs, put them in a co-culture system for a couple of days, and look for CD8 T cell activation. Another form of the assay is putting the peptides in crude PBMCs, and this goes pretty fast. You're able to screen about 100 peptides in 3 months' time. You can also work with patient PBMCs, which are sometimes hard to get in large volumes. So using these two assays, we did our proof of concept screen about 100 peptides, and we found a superior accuracy in predicting immunogenic and non-immunogenic peptides using the OncoPepVac output. One interesting thing that we noticed during our assay is that a significant fraction of these peptides actually elicit an inhibitory response with T cells. That instead of activating and producing interferon, they actually inhibit interferon production by T cells. And this is not done in one donor. Most of these assays were done in 2 to 5 donors. So here is a positive peptide where you see positive responses in multiple donors. And here are the series of peptides that actually suppress interferon response. So this data then really highlights that running a screen post-prediction is important, because you would want to parse out these peptides that would actually inhibit your T cell activity rather than activating your T cells in vivo. The other interesting thing we noticed is there is a huge variation in the positive peptide response between the different donors. And we decided to take a closer look at that, and one of the reasons we think that this could be is that we saw there's a large variation in HLA expression in different donors. On the left, you can see is HLA A, B, and C expression in different cancer, a series of cancer patients, and they are expressed at different levels. Now we know that in cancer there is immune escape and there is HLA expression gets suppressed, but also in normal donors, healthy donors on the right, we also see a large variation in HLA expression. So this could be the first step, that is, if antigen presentation is impacted, you would of course land up with variability in your assay. Other problems we encountered was there was differences in monocyte maturation into DCs. Also, PBMCs as such are very costly if you want to run these assays in triplicates. It's hard to find rare HLAs. With regards to patient PBMCs, we get them in very low amounts with very poor viability. And also, if you are looking for an HLA-specific response, testing them in PBMCs is challenging because you have 6 alleles of HLA. 6 alleles of HLA being expressed. So in order to overcome these obstacles, we decided to engineer an artificial APC which would be a renewable cell capable of presenting the antigen and would express T-cell co-stimulatory molecules. So in this case, we would just express one HLA and the cell will not express any endogenous HLA. So then you're really querying the HLA-restricted peptide presentation and assaying its antigenic potential. We are putting these peptides on either exogenously or endogenously using mini genes. When we put these cells in culture with peptide and T cells, this is some of our preliminary data where the green bars show a response. This is on the y-axis, you're looking at interferon response from CD8 T cells. The green bar here shows a response to flu antigen, so there's a mix of 32 flu viral peptides. And the gray bar here is melena, which is a known cancer antigen. So after the first re-stimulation, you can see there's when you have a potent mix of antigens, you start seeing a response. But with regards to cancer antigens, the response starts showing at a much later time point. Currently, we are also developing a mini-gene-based vector delivery system, and these are some of our preliminary experiments where we are putting in the antigen internally rather than adding them as peptides from outside. And the advantage of that is sometimes if you just test peptides as 9-mers, they're not going through the regular processing machinery, and you might land up getting false positives. So once you put in your gene of interest internally, then that's a higher bar for the peptide to cause T-cell activation. But the downside of it is it's not a high throughput system. Additionally, we are also set up for looking at identifying new antigens based on mass spec-based method, where you IP HLA peptide complexes, you assay the peptides, and you can actually identify a couple hundreds of 9-mer peptides. Most of them are wild type, and some of them are mutant, which would be your antigens that are at the surface of the cell. Now, so far I've talked about peptides eliciting T-cell activation and interferon response. Next, I'm going to switch gears and talk about peptides eliciting a T-cell response with regards to change in TCR repertoire. So TCRs are consisted of two chains, the alpha chain and the beta chain. And the CDR3 region of the alpha and the beta chain dictate the diversity dictate the specificity of binding to the peptide antigen. And these CDR3s, there's a large repertoire present in endogenously, anywhere in the range from 10 to the power 6 to 10 to the power 11. And so when a TCR sees an antigen, it undergoes clonal expansion, and the percentage of the particular CDR3 gets enriched in the repertoire. So when we run a TCR analysis on our assays, you can actually detect antigen-specific TCR clones. What you're looking here, each dot here is a unique CDR3 sequence. And you can see that the dots along these lines are the TCRs that are unique to the antigen compared to the DMSO control. And in particular, this particular sequence shows an enrichment of up to over 50%. So here is one case where we see unique enrichment of one TCR clone. Since we have screened hundreds and hundreds of peptides, we see multiple types of TCR expansion, TCR clonal expansion in our assays. Certain peptides elicit expansion of one clone, whereas in other peptides you see expansion of more than one clone. And we have been able to achieve this using Takara's Smarter TCR profiling kit and also 10x's VDJ sequencing kit. Now, one thing that did strike us as we were running these assays is that TCR expansion was not always correlated with interferon gamma production. And that made us think that when you have a polyclonal TCR response, how many of them really do have a functional effect on the TCR cell? So in order to do that, we set up an antigen presenting assay, and after antigen CD8 T cell activation assay, and at the end of the assay period, we channeled our cells through 10x's single cell sequencing platform, where you're looking at TCR expansion coupled to transcriptome profiling. When we did that, we see that here, for example, at the TSNE plot, 3 TSNE plots showing all TCR clones in light blue that were expanded. The dots in dark blue are the unique clones that have expanded in response to the antigen into significant amount. And what you can see is that clone 1, clone 2, clone 3 produce varying amounts of interferon gamma. So not all T cells that are expanded are actually functional. So that was an interesting insight for us to see that effect. Now moving on to in vivo TCR profiling, which can be quite informative if you are looking for shared antigens across tumors. But the challenge is that most of the tumor tissues that are available are FFPE, and you have very small amounts of material to work with. So we used some of Takara's kits and modified the protocol, and we were able to set up successfully amplified TILs from FFPE tumor tissues. And here, this is just to give you an idea of what a normal RNA profile looks like. Here's the 18S and the 28S, and this is what the RNA from a degraded FFPE sample, degraded RNA from FFPE samples look like. So for these RNA, we use a criteria that's called the DV200, which is the fraction fraction length of your RNA that's greater than 200 base pair. So for a normal sample, it's close to 98%, but for an FFPE sample, it could be as low as 20%. So working with this in order to get the TCR information could be quite challenging. So here is some of our initial data looking at TCRs from tumor samples, and this would be then looking at TILs in the tumor sample. So these are the different TCR sequences, and this is their clonal enrichment. So this is a PBMC sample, and you can see that TCR clones are enriched to a certain percentage. But when you go into a tumor, you see large amplifications of particular TCR clones. And in fact, these two are the same tumor types, and the top 2 clonotypes show a match, which could potentially mean that we are looking at shared antigen-induced TCR clones. So this is indeed quite exciting, and we continue to use some of our own, use these resources for our own research, and also it's out there for the general community to use. So we do offer it as a service to the biotech community. In addition, we have high capacity NGS capabilities. We have the NovoSeq, HiSeq, Extend 2500, MySeq, and the 10x platforms. We offer multiple data analysis capabilities, including TCR repertoire analysis. And so to summarize, these are the various service offerings we have. We have a robust sequencing platform where we can meet your sequencing needs for your specific tissue. OncoPepVac, the vaccine prediction pipeline that I talked about, using TCR binding as a criteria for antigen selection, helps you sequence, predict antigens with high confidence. The OncoPep screen assay, which will help you validate those antigens further. And then our TCR sequencing platform that will help you look at the TCR-specific response to those antigens before you decide to push them in as a vaccine cocktail. And that's it for today. Thank you for your time.